Hey, this is the video for Diffusion.1. So go ahead, set up your Cornell notes, start a new entry by completing your setup. Pay attention to the essential vocabulary. We will be looking at the words diffusion, passive transport, and concentration. So one quick analogy. You've all taken the subway. Some of you take subway to school. The L train especially is quite crowded in the mornings and in the afternoon. And you notice that sometimes you have a very crowded train, but a nearly empty platform. So my question to you, is it easier to get onto a crowded train from the empty platform? Or is it easier to get off of the crowded train onto the empty platform? What's your response? So this very much represents what happens with diffusion. So imagine you have a beaker of water and you're adding a drop of food coloring. You've probably noticed, so the food coloring enters the water and you don't really have to do anything, and eventually that food coloring will fill the entire container. This is what happens in diffusion. So our first vocabulary word, diffusion is the movement of particles from areas of higher concentration to areas of lower concentration. When that drop went in, the concentration of red food coloring was very high where there was a drop. Where there was just regular water, the concentration was low. So this food coloring spread from the high concentration area to the low concentration area. Diffusion is passive transport because there is no energy involved. And in fact, diffusion and passive transport mean the same thing. They'll be used synonymously when you see Regents questions. And the last thing that I would like you to do in your notes is to sketch a diagram. So have some dots here where they're close together, an arrow, and then dots that are farther apart. This represents high concentration. This area represents low concentration. It is spreading out. So now when we're talking about particle size, along our cells there are small little openings, and some particles are very small, like glucose and water. But some particles are actually quite large, like polymers, especially starch. Remember that starch is a bunch, and there's many, many more than this, but starch has hundreds or even thousands of glucose molecules bonded together. So what you notice is that both water and glucose can enter the cell. But starch is too large. It cannot enter the cell. And you saw this when you did the diffusion through a membrane lab. The diffusion through a membrane lab used a plastic bag with little tiny microscopic holes that you can't see. And you put starch and water and glucose on the inside of this bag. And you put iodine on the outside. And afterwards, you noticed that the starch stayed inside because the starch changes into a blue-black or a dark purple color when it is exposed to iodine. Remember, iodine is an indicator. So the iodine was actually small enough to go into this membrane, but the starch was too large to leave the membrane. This was one of the big takeaways from this lab. And you will be asked questions on the Regents, especially in Part D. So what I would like you to do right now is complete this check for understanding. And I hope that this video was informative. I thank you for your attention. I will see you soon.